My name is Taryn, and I'm a member of the SI Bridges program. Today, I'm going to give you a short overview of my poster presentation for summer of 2022. I currently work with Dr. Young, and we look to see if mitochondrial DNA can be used as a blood-based biomarker for endometrial cancer. So I'm going to zoom in on the poster here just a little bit so you can get a better look at what's going on here. So as a little introduction, endometrial cancer, also known as EC, had 417,000 new cases in 2020 and is the 6th most common cancer in women and the 15th most common cancer overall. It occurs in the endometrium, as you can see here, the mouse. We have a little diagram, and this little blue ball right here is where the endometrial cancer is. So what happens when we have cancer? So we have healthy cells which grow, multiply, and die at a set rate. But these can mutate into abnormal cells which grow and multiply, but they don't die at a set rate, and they can also multiply uncontrollably. And the buildup of those abnormal cells is what creates a mass or a tumor. And we also know that damaged or defective DNA has the ability to change those important genes that can control that cell growth. And when those genes are damaged, that can cause cancer. So we talked a little bit about mitochondrial DNA, so that is what we looked at, and here's a little diagram of what mitochondrial DNA looks like. We have pointed out all of the different mutations in endometrial cancer patients that we've seen so far. So going into methods, we did PCR, which in a nutshell, what that does is it multiplies the amount of DNA that we have. We also did gel electrophoresis, which helps us measure how much we have. If you are interested in how these processes work, they are very fascinating. I would highly recommend the PCR and gel electrophoresis videos from the Amoeba sisters. They are fun, easy to understand, and it makes science really fascinating. We also did gel extraction, which essentially we take the DNA that we have in our gel during our electrophoresis, and we separate the gel from the DNA. So we have just our DNA again. And we did quantification last, which just tells us how much DNA we have. It helps us measure it. So over here, we have a little example of what a gel looks like. So that is what the end of gel looks like. These little bars are the DNA. Moving on to results, this is a table, a little summary of the work. So we have our patient and mutation over here, the primers that we used, the amount of template that we used, the amplicon concentration, and the total amount of DNA that we have at the end. Over here on the end, we also have the primers that we used. And after we finished everything, we sent off the samples for next generation sequencing with our collaborator. And this next generation amplicon sequencing is what will detect the mutations of the mitochondrial DNA. So on to discussions. We're still currently waiting for the results. But the results from this research can present a possible non-invasive testing option for those with endometrial cancer. So the overall big picture of this research project is looking to see if we can diagnose endometrial cancer by a simple blood test. So whenever we take blood, we would be looking for those mitochondrial mutations in the blood serum specifically. So that is a big picture of this research poster, and we are hoping that if results come back and we can detect it in our blood, we might be able to possibly use that to expand and be able to detect other types of cancer with this technique or something very similar. This would be extremely helpful not only for those with other types of cancer, but also endometrial cancer, of course, because this could mean catching cancer that is recurred earlier. EC is most likely to recur in the first few years after treatment, and certain types of EC and cancer treatments have been connected to them being at an elevated risk for other cancers such as breast, colon, rectal, lung, lymphoma, bladder, kidney, soft tissue, etc. So fingers crossed that we'll hopefully get results soon. And lastly, I would like to acknowledge and thank the Simmons Cancer Institute at SIU and the SIU School of Medicine for supporting this research, as well as SI Bridges, Dr. Young, of course, William Browning, Pabitra Kadka, Carolyn Young, Mostafi Juraman, and Lakshmi Sagwan. Thank you all for your support, and hopefully we'll see you next summer. Thanks.